Alright, let's talk about DNA barcoding, PCR, and sequencing. First, let's define a few terms. When we refer to the genome of an organism, it's all of the DNA that resides inside of it. It includes chromosomes, which have the majority of the genes that control your phenotype, as well as DNA from mitochondria, and if you're a plant, things called chloroplasts. Mitochondria and chloroplasts have their own DNA, which is circular, looks a lot like bacterial DNA. A human genome is about 3 billion DNA letters long, or base pairs. A fungal genome is, on average, about 50 million base pairs long. That includes all the information to make all the proteins, enzymes, and other components of the living organism. On the other hand, a DNA barcode is just a very tiny piece of the genome, maybe a few hundred base pairs long. What makes a DNA barcode special is it doesn't tell us anything about the organism itself. You wouldn't be able to tell that a mushroom was blue or whether a mushroom was poisonous just by looking at a DNA barcode. The DNA sequence within it is mostly arbitrary for our purposes. What we're doing is comparing the same region of the DNA between two different species. And a good DNA barcode changes just often enough that there are differences between the DNA barcode for two different species. In fungi, the most common barcode used for species level identification is called the ITS. And ITS stands for Internal Transcribed Spacer. It's a region that actually includes two components that we refer to as ITS1 and ITS2. When we sequence the ITS region, we're going all the way from the small subunit to the large subunit, and including the space in between them, which also has the 5.8s. If you recall, these are components of the ribosome. So the small subunit is also known as the 18S in eukaryotes. Then there's the 5.8s, and then there is the large subunit. This component of the genome that we call the ribosomal RNA cistron is really good for barcoding because it actually has many copies in the genome. A typical gene will have two copies, one from each parent in a diploid organism or a dikaryotic fungus, whereas this is actually repeated many times. And it's because it's so important, it needs to have many copies, it needs to be redundant, and it needs to have a lot of transcription happening on it. So unlike regular genomic DNA that would be, let's say it's a gene and it's transcribed into messenger RNA, this RNA is not messenger RNA. When this region of the genome is transcribed into RNA, it's called ribosomal RNA. And it doesn't undergo translation and become a protein or a gene product. This RNA is actually structural. So when it's transcribed, it will fold up and mix with some proteins and become the components of the ribosome. The internal transcribed spacer is the part of this that separates the components of the ribosome. And so we have ITS1 and ITS2. The DNA sequence in these regions tends to be very variable. And that's because by themselves they don't have a particular function. The only function that these two regions need to have is the ability to cut themselves out after they are transcribed so that the different components of the ribosome are separate. The red components here, the parts of the ribosome, are extremely well conserved in eukaryotes. And so we target this region because you can create a primer sequence that works for large categories of eukaryotes, or you can make them a little bit more specific. So for our ITS sequencing, we use a forward primer called ITS1F, and the F stands for fungal specific. It amplifies most fungi. The reverse primer is ITS4, and that has U in parentheses. The U refers to universal. So the universal primer means that it's going to work in almost all eukaryotes. So together we capture a little bit of the small subunit, all of ITS1, the entire 5.8S, and all of ITS2. So when we're going to go sequence this region, we don't just sequence the entire genome and pull this out, although you can do that. We want to take advantage of the fact that there's many copies of it, and we don't need a lot of DNA to start out with to make many copies of just the sequence that we care about. So to do that, we use a process called PCR, which stands for Polymerase Chain Reaction. Polymerase being the DNA enzyme. And so our objective is to make billions of copies of the DNA barcode sequence. There's a bit of a problem in that the machinery inside of our cells that copies DNA is incredibly complex. There are many enzymes and proteins and other components that are required to successfully copy our DNA. And to be able to recreate that process in a lab condition is extremely challenging and expensive. So one way around using this huge suite of enzymes that can unzip the DNA and prevent it from supercoiling is kind of hacking the chemistry of DNA. The nitrogenous bases are held together with hydrogen bonds, which are fairly weak in the sense that it doesn't take a lot of energy to separate those two strands of DNA. What we can do is we can heat up the DNA, and once we reach about the boiling point of water, or about 94, 95 degrees Celsius, that DNA unzips. Those hydrogen bonds collapse, and you end up with single strands. And with a single strand, you're able to attach those primers as bookends and copy the DNA. The problem is that the enzyme that we use in our body and most of the enzymes that do this basically fall apart once you get to the temperature required to unzip the DNA. So the solution to that problem was isolating a DNA copying enzyme called Taq polymerase from a bacterium called Thermus aquaticus. It was originally isolated from a hot spring called Mushroom Spring in Yellowstone National Park. Don't think it's much of a coincidence, do you? Okay, so once we have these ingredients in place, we can do our PCR. And the ingredients are our DNA template. So that is the genomic DNA that we've extracted from a fungus, all of their DNA. Then we need the DNA primers, and those are going to serve as bookends to basically cut out the DNA sequence that we want to make many copies of. Then we need DNTPs. These are the building blocks, the individual nucleotides. A enzyme, that's the Taq polymerase, and a buffer solution that keeps the pH of this reaction fairly constant. So in order to run PCR, we need a device called a thermal cycler, and it does exactly what it sounds like. It changes temperatures in a cycle. So the first step of PCR is melting that DNA. It's raising the temperature, allowing the two strands to separate. The next process that we do is annealing. This is where we cool down the temperature just a little bit, and our primers will stick to their complementary sequence and serve as bookends for the thing that we want to copy. And then during a different part of this thermal cycle, where we change the temperature again, it's the temperature at which the enzyme is most active and will actually copy the DNA. At the end of a cycle, we've got sequences that are copies of only the region that we want. And then after 2 cycles, we end up with more copies. And as you go through each cycle, the amplification is exponential. So you go from 2 to 4 to 8 to 16 to 32, etc. So this is a schematic of the ribosomal ITS region, and so here we can kind of zoom in and see where the primers anneal. So this ITS1F primer, which has the forward arrow there, we have a primer that sticks there. The ITS4 primer goes in the opposite direction. We have a primer that sticks to the other strand, and then we amplify the region in the middle. We'll just walk back through this again. So the first stage is denaturation. That's where the DNA unzips at about 95 degrees Celsius. And this is when our primers can attach. At about 55 degrees Celsius is when our primers can reattach, but the DNA still stays separate. At 72 degrees is the optimal temperature for the Taq polymerase to copy the DNA. And then we repeat that about 35 times. Once we have our billions of copies of DNA, there's a couple of different ways we can sequence that and actually get the DNA barcode. So the old school method is called Sanger sequencing or capillary sequencing. In order to do this, you take your billions of copies of DNA and then you do another round of PCR. But instead of adding only just regular DNA letters, every so often, 1 out of every 500 of these DNA letters is a special one called a terminator, or a dideoxynucleotide triphosphate. And each of these different letters has a color associated with it. A little component called the fluorophore that, when excited by a laser, will show up as a particular color. When one of these is incorporated during PCR, during that copying, it will actually stop the copying process. And you'll end up with a strand of DNA. Let's say it's this strand right here, and it'll add nucleotides, add nucleotides, add nucleotides, keep extending until one of these special ones comes into place, and then it stops extending. And so you end up with a fragment that ends in a fluorescent nucleotide. When you do this, and you have billions of DNA molecules, and you've got, hundreds of billions of these things floating around, you end up generating fragments of DNA of every possible size of the entire DNA sequence, and each of them will end in one of these special nucleotides that turns fluorescent when a laser scans it. So let's go over this again. So we start out with our PCR product, which now we're calling template again. It's what we're starting a new round of PCR with. Here's our dideoxy nucleoside triphosphates and all the other components. And so we do our PCR. One of these rare nucleotides gets added, and you end up with a fragment that ends in one of these fluorescent nucleotides. You end up with fragments of every possible size range just by random chance. Then you take these and you run them through a very thin capillary tube that's filled with gel. And you run them through with an electric current, and the bigger pieces of DNA running through that gel will run through a lot slower because the gel is this thick matrix that impedes their progress. The small pieces of DNA are not being impeded by the gel and will move through quicker. By the time you get to the end of this capillary tube, you've had just enough separation of these different size fragments that the first thing to come across this laser is going to be the shortest fragment, and the last thing would be the longest fragment. And in between that, you'll have fragments of every single size ending in every possible nucleotide in that sequence. So as it comes through, this would read T, A, G, C, T, G. Now, these are not perfect examples because it's not exactly one letter longer, but if you get the picture, it scans these as they come through, and you end up with something called a chromatogram, and that shows you the sequence that the DNA passed by that laser. And it shows you in the fluorescence intensity. So that's shown as these peaks, and each of these peaks corresponds to one of these fluorescent nucleotides coming through. So this is old school. You can't really generate a lot of data this way, but the data you do generate is pretty robust, and you can look at the raw signal and interpret that, although a computer program will read it for you. You just have to double check it sometimes. The next step up from this is what was called next generation sequencing. We can also call it second generation sequencing. And it's an entirely different technology. I'm not going to get into the details of how it works, other than it has vastly larger throughput. So throughput meaning you can generate far more data at once. Sanger sequencing is the technology that was used to sequence the human genome, and it took about 10 years to sequence 3 billion base pairs and stick them all together into a whole genome. The max output from a single run, you can maybe get DNA sequences that are about 1,500 base pairs long, although in practice this is kind of the upper limit. And you can do about 384 samples in one run. So with a total yield or total throughput of about 600,000 base pairs, give or take. When you're using the next generation sequencing, which is commonly Illumina technology, you can generate trillions of base pairs. So the maximum output of one of the more recent Illumina sequencing machines called the NovaSeq 6000 is per flow cell you can generate 3 trillion DNA letters worth of data. That is many more orders of magnitude larger, and you can sequence an entire human genome 1,000 times over. So pretty awesome. The applications for this technology are beyond DNA barcoding, and you generate so much data that you're really using this for either whole genome sequencing, meta barcoding, eDNA sequencing, or very large-scale DNA barcoding when you want to generate millions and millions and millions of sequences. The disadvantage to this technology, well, first of all, it's extremely expensive. One of these machines is like $1 million, and then there's a very pricey service contract and proprietary reagents. It's not something that any individual can really afford, and the only thing that makes this machine worthwhile is if you are running sequencing all day, every day, massive data sets. The other disadvantage is that the length of the reads that it generates is short. So with Sanger sequencing, each read can be about 1,500 base pairs maximum. With this, the maximum size is 250 base pairs, although most commonly people generate 150 base pair sequences. So a typical fungal ITS region is going to be about 650, 700 base pairs long. You can't capture that in a single read. So when you sequence with this, you have to either go for sequencing this region here, you want to get ITS1, or you want to get ITS2. You can't do both. Okay. The newest player in the game is third generation sequencing. The advantages here are startup cost under $15,000 to set up a lab at home with all the necessary equipment and supplies. Sure beats $1 million, and there's no service contract. The consumables, while expensive, you don't need to buy large quantities of. This can enable barcode sequencing for less than $1 per specimen, in many cases much less than $1 per specimen. It's extremely portable. So here's an astronaut in space using the DNA sequencer right there. So if you recall, this thing, in reality, this is like the size of like the biggest printer, copier, fax machine you've ever seen. It's huge. This, on the other hand, is this tiny little device here, plugged into a tablet. The rest of the equipment you need to run this can fit into a suitcase. This device is extremely flexible. So you can do short read sequencing, long read sequencing. It has a theoretical capacity to sequence reads that are up to and over 1 million DNA letters long by itself. You can also do RNA sequencing. So for mRNA or other types of RNA, you can sequence that directly without having to generate cDNA. That's another topic. So we use this particular third generation sequencing technology that is called the Oxford Nanopore Technologies MinION sequencer, and it uses nanopore sequencing technology. There's another third generation sequencer that can do long reads. That's called the PacBio, and that is more like the Illumina unit, where it's extremely expensive, it's very large, and is really only useful if you are using it to generate massive amounts of data. So here's a picture of the sequencer in action. This is in Hawaii, and you can see the device here just plugged into a laptop. It's pretty awesome. So let's talk about how the sequencing works here. So we went over Sanger sequencing. This is entirely different. We're directly sequencing the DNA. We're actually looking at the DNA without making copies of it, without adding weird fluorescent stuff to it. And the way this works is there's a biological molecule. It's a type of protein called a porin, and it exists in bacteria. Specifically, the one that inspired this sequencing technology came from Staph aureus, which is one that you've probably heard of. There's a virulent one called MRSA, MRSA, and that has a protein called alpha hemolysin. Hemolysin meaning breaking of the blood. So the bacteria produces this protein, kind of looks like a mushroom, and it inserts itself into biological membranes. And it does, in humans, it does this to our red blood cells and breaks them open and leaks the contents out. But someone was really thinking pretty hard about this and realized that the dimension of this tube in the middle of this protein, this is a two-dimensional representation. It's really here's a better sort of 3D look at it. It's an actual pore here. It's about the same size as a single strand of DNA. So that DNA that's been separated into two strands. And so over time and with lots of development, this nanopore sequencing technology has come about where these proteins are embedded in a membrane in an electronic device, and this membrane separates two electrolytes, and there's a different electrical potential across the membrane. So you have a concentration gradient such that when you allow ions to move from one side to the other, you end up with an electric current. And so by stuffing these pores with single-stranded DNA and shoving them through, with each DNA letter comes a different number of ions across this membrane, creating an electrical current which can be measured at the pore. So each DNA letter can carry a different amount of this current with it. And by reading that, you can sort of estimate the DNA signal. Now, it's not reading each single DNA letter at a time. It's reading a couple of them. And so the signal is kind of averaged out, and it takes a really, really complex computer program to solve that electrical signal and turn it into DNA letters. It's not like looking at a raw signal output from a laser where you can see a peak of fluorescence. This is far more complicated. But we're directly reading the DNA as it's flowing through the pore, which is pretty exciting. The results that we get from sequencing can then be interpreted so that here's that Sanger chromatogram that comes out where it shows you the peaks in the letters, and the output from nanopore sequencing would be a bunch of individual reads of that same DNA sequence. And critically, you have some error that happens in any sort of sequencing technology. But when you look at a nanopore sequence, you need to make a consensus of many sequences to make sure that any errors are accounted for. And you can see an example of that here where you've got some random error, but it's only in a few sequences, and it seems to be in a basically random position every time. So if you get the consensus of all these different sequences, you end up with the correct sequence. And that's more or less what's actually happening with Sanger sequencing. It's not like a single DNA molecule comes through that capillary tube at a time. You're getting, thousands, millions of DNA molecules of the same size traveling together through that gel. So this is also a consensus, because even within the DNA, there are errors. Polymerase that we use for PCR makes about one error in every 5,000 because it's not nearly as accurate as human DNA replication. There's other sources of error as well. There's also some variation in the DNA sequence between the different copies of this ribosomal RNA. So in any case, we're going to have to use some interpretation to look at this data and make sure that it's accurate. When we have the DNA sequence, we can compare it against millions of other sequences using a blast search. We're going to get into that quite a bit more in detail as we go through this program, but this is a representation of comparing two DNA sequences using a blast search. And you can highlight the areas where those two sequences don't match each other. Okay,